we'll be looking at using CRISPR as a possible therapeutic treatment for SARS-CoV-2. SARS-CoV-2 stands for Severe Acute Respiratory Syndrome Coronavirus 2, and as we know, this virus has been spreading to the human population as of December 2019. A coronavirus is a virus whose genetic material is coded in RNA, and the main defining feature of these viruses is that the spike proteins found in them is shaped like a crown. This picture here puts emphasis on the shape of that spike protein being shaped like a crown, as it plays an important role in how this treatment was developed, and also plays an important role in how the virus is able to infect our cells. In red, we can see the spike proteins found all over the cell, and in gray, we can see the cell membrane. Inside the cell membrane of the virus, we can find viral RNA, and we can also find enzymes able to transcribe that viral RNA. When we say transcribe, we mean that it's able to change that viral RNA into a form that can be identified by cell machinery to make proteins. I'm going to see how that process would affect us inside of our cells. Top left, we see that the virus will approach the cell and use the spike proteins to bind with receptors on your cell, allowing it access into your cell. Now that it has access, the viral RNA will be inside your cell and then proceed to make copies of itself and transcribe itself. It will then transcribe itself into mRNA here, which can then be identified by the cell machinery of your cells to make the molecules that will make up new viruses, which will essentially turn your cell into a virus factory. Now we understand that, we move on to what is CRISPR real quick. CRISPR is a bacterial immunological defense system that can only be activated after a bacteria survives a viral infection. The reason why this can only happen afterwards is because we need a template to work with. Essentially, CRISPR-associated proteins need a template to work with, so it can only occur after the bacteria survives a viral infection. We also need a target sample, which would be a reinfection in case of a bacterial. The most famous Cas protein is known as Cas9 because this protein has been used by scientists over the years for genetic editing of various organisms, and it's also because of how specified these Cas proteins really are. Pink, we can see the Cas protein, and in his left hand, we can see that he has a template, and around it is various DNA molecules that would then be compared to the template to see if it will be a match. When a match is found, the CRISPR-Cas protein will be activated, and it will then proceed to cut that segment out of the DNA. In the case of a bacteria, that would just kill the virus that's trying to invade. In the case of organisms, it usually shuts off genes. Now we see how the bacteria is able to save a template. So in this case right here, we say that this is a healthy bacteria. We just have the DNA inside. Then we get infected. We have to open up that DNA so that we can transcribe it to make the proteins necessary for cell repair. While it's doing that, it will also incorporate a segment of the viral DNA that's floating around into its own DNA to save it in an archive so it can be used later as a template for the CRISPR-Cas system. So now we have established that coronaviruses use RNA to replicate, and we established that Cas9 uses DNA to replicate. So obviously we can't use Cas9 as a way to treat RNA viruses. This is where Cas13a comes in, because Cas13a, unlike Cas9, is able to cut RNA. And also, when activated, unlike Cas9, which just cuts the DNA and then that's it, Cas13a goes into collateral cleavage mode, which then it becomes unspecific, unlike how it originally was. It will only cut what it found. It will cut anything it comes by that's RNA. Now we see how that will work inside the cell. In the blue circle, we can see that we have the viral RNA. The CRISPR-Cas13 system will come by, identify the match to the template it has, and then be activated. Once activated, it will cut that viral RNA inside the blue circle, then go into collateral cleavage mode, and then proceed to cut all the mRNAs that are inside the red circle. This will then lower the production of new virus particles, effectively lowering the viral load of cells. Would this treatment be safe is the question you might be thinking now. Now, this treatment is very safe because the scientists who designed it went through a lot of steps to ensure that the specificity of the CRISPR protein was very high. The way they were able to do this is by comparing the RBD of SARS-CoV-1 with SARS-CoV-2, which most people don't know. This is not the first coronavirus to infect humans, and this is actually very common to one that infected people in China in 2002. So I mentioned the RBD. The RBD is receptor binding domain. This is a section of the viral RNA that codes for the spike protein. So it has the instructions for how to make spike proteins for the virus. They compared that of SARS-CoV-1 and SARS-CoV-2, and it's actually very different. So this is what they use as the template to make the CRISPR RNA that the CRISPR-Cas protein would use. How they did this is by using 3D modeling and by running molecular docking simulations of how the CRISPR Cas protein would fit with the CRISPR RNA template. They made 39 templates to match that RBD of SARS-CoV-2, and they narrowed it down to CRISPR RNA-6 being the sixth template they made to be the one that was best effective at binding to the RBD of SARS-CoV-2. Now, you might be thinking, who is this treatment really targeted for? Because I said it was therapeutic. So what that means is that this treatment would be best for people who already have the virus, i.e. being therapeutic, but it wouldn't be for people in the later stages of it because of that collateral cleavage effect. It would actually be best for people who have been recently diagnosed with coronavirus, so CoV-2. This is because they will have less cells that have been affected, which would mean we have less collateral damage because that collateral cleavage effect is going to cleave any RNA it finds. How would this treatment be given to patients? Is another thing you might be thinking. And the people designed this treatment did not have a guaranteed safe way to distribute this to people because it's actually quite complicated in how to get this CRISPR-Cas system into the cells of a person.